Welcome to our exploration of mutations and polymorphisms. Before we dive into the details, let's set the stage by understanding the basics of genetics. Our DNA is like a blueprint, carrying instructions that determine everything from our eye color to how our bodies function. These instructions are encoded in genes, which are segments of DNA. Sometimes changes can occur in these genetic instructions, and that's where mutations and polymorphisms come into play. In this video, we'll break down what these terms mean, how they arise, and why they matter in biology and medicine. Let's begin our journey into the fascinating world of genetic variation. Mutations are changes in the DNA sequence that can occur naturally or be induced by environmental factors. These changes might be as small as a single letter in the genetic code or as large as entire sections of DNA being rearranged. Mutations can happen during DNA replication, when cells divide, or due to exposure to things like radiation or certain chemicals. Not all mutations are harmful. Some have no effect, while others can lead to diseases or even beneficial traits. Understanding mutations is key to grasping how genetic diversity arises and how certain conditions develop. There are several types of mutations, each affecting DNA in different ways. Point mutations involve a single nucleotide change, while insertions and deletions add or remove nucleotides from the sequence. Larger mutations, like duplications or inversions, can impact bigger sections of DNA. Each type of mutation can have varying effects on gene function depending on where and how they occur. Some mutations might disrupt a gene entirely, while others might have subtle or no effects at all. Recognizing these types helps scientists predict potential outcomes and understand genetic diseases. Mutations can arise from a variety of sources. Some are spontaneous, occurring naturally as cells divide and DNA is copied. Others are induced by environmental factors such as ultraviolet light radiation or chemicals that damage DNA. Even errors during DNA repair processes can introduce mutations. The likelihood of mutations increases with exposure to mutagens, which are agents that cause genetic changes. By understanding the causes, researchers can better assess risks and develop strategies to prevent harmful mutations, especially those linked to diseases like cancer. Effects of mutations can range from harmless to life-altering. Some mutations are silent, meaning they don't change the function of the gene at all. Others can cause genetic disorders or increase susceptibility to diseases. Occasionally, mutations can be beneficial, providing advantages that help an organism survive and reproduce. For example, certain mutations have led to resistance against diseases or adaptation to new environments. The impact of a mutation depends on its type, location, and whether it affects critical genes. Mutations play a crucial role in evolution by introducing new genetic variations. Over generations, these variations can accumulate and lead to the development of new traits or even new species. Natural selection acts on these mutations, favoring those that provide a survival advantage. Without mutations, evolution would not be possible, as there would be no new traits for natural selection to act upon. This process explains the incredible diversity of life we see on Earth today. Polymorphisms are a specific type of genetic variation that occurs commonly within a population. Unlike rare mutations, polymorphisms are present in at least 1% of the population. These variations can be as simple as a single nucleotide change, known as single nucleotide polymorphisms or SNPs, or involve larger segments of DNA. Polymorphisms contribute to the diversity seen among individuals, such as differences in appearance, blood types, or even how we respond to medications. Both mutations and polymorphisms involve changes in DNA. The key difference lies in their frequency and impact. Mutations are often rare and can have significant effects, sometimes causing diseases. Polymorphisms, on the other hand, are common and usually do not cause disease. Instead, they contribute to normal genetic diversity within a population. Understanding this distinction helps scientists study genetic diseases and population genetics more effectively. Single nucleotide polymorphisms, or SNPs, are the most common type of genetic variation among people. Each SNP represents a difference in a single DNA building block called a nucleotide. SNPs occur throughout the genome and can influence how individuals develop diseases or respond to drugs. Researchers use SNPs to study genetic predispositions and to personalize medical treatments, making them a powerful tool in modern genetics. Besides SNPs, there are other types of polymorphisms, such as insertions, deletions, and copy number variations. These changes can involve larger segments of DNA and may affect gene function or regulation. Some polymorphisms are associated with specific traits, like lactose tolerance or resistance to certain diseases. By studying these variations, scientists gain insights into human evolution, health, and disease susceptibility. Detecting mutations involves various laboratory techniques. DNA sequencing is the most common method, allowing scientists to read the exact order of nucleotides in a gene. Other methods include PCR polymerase chain reaction, which amplifies specific DNA segments, and microarrays, which can screen for many mutations at once. These technologies have revolutionized genetics, making it easier to identify mutations linked to diseases or traits. Polymorphisms are detected using similar techniques as mutations, such as DNA sequencing and microarrays. Additionally, restriction fragment length polymorphism (RFLP) analysis can identify specific DNA variations. These methods help researchers map genetic diversity within populations and understand how certain polymorphisms influence health and disease. The ability to detect polymorphisms has opened new doors in personalized medicine and ancestry research. Medicine, understanding mutations is crucial for diagnosing and treating genetic disorders. Some mutations cause inherited diseases like cystic fibrosis or sickle cell anemia. By identifying these mutations, doctors can provide early diagnosis, genetic counseling, and targeted therapies. Advances in gene editing, such as CRISPR, offer hope for correcting harmful mutations and treating previously incurable conditions. Polymorphisms also play a significant role in medicine, particularly in pharmacogenomics, the study of how genes affect a person's response to drugs. Certain polymorphisms can influence how individuals metabolize medications, making some drugs more or less effective or increasing the risk of side effects. By understanding these genetic differences, doctors can tailor treatments to each patient, improving outcomes and reducing adverse reactions. Many cancers are driven by mutations that disrupt normal cell growth and division. These mutations can be inherited or acquired during a person's lifetime. By studying the specific mutations involved in cancer, researchers can develop targeted therapies that attack cancer cells while sparing healthy ones. Early detection of cancer-related mutations can also improve prognosis and guide treatment decisions. Polymorphisms can influence an individual's risk of developing certain diseases. For example, some SNPs are associated with increased risk for conditions like diabetes, heart disease, or Alzheimer's. However, having a risk-associated polymorphism does not guarantee disease. It simply means the risk is higher compared to others. Lifestyle and environmental factors also play a significant role. Understanding these genetic risk factors helps in prevention and early intervention. Mutations can be classified as inherited or acquired. Inherited mutations are passed from parents to offspring and are present in every cell of the body. Acquired mutations, on the other hand, occur during a person's lifetime and are not passed on to children. These can result from environmental exposures or errors in DNA replication. Both types are important in understanding genetic diseases and cancer. Germline mutations occur in reproductive cells and can be passed to offspring, affecting future generations. Somatic mutations, however, occur in non-reproductive cells and are not inherited. Somatic mutations can lead to diseases like cancer, but do not affect a person's children. This distinction is crucial for understanding how genetic changes are transmitted and their implications for health. Not all mutations have noticeable effects. Neutral mutations do not affect an organism's fitness or health. They may occur in non-coding regions of DNA or result in changes that do not alter protein function. Over time, neutral mutations can accumulate and serve as markers for studying evolutionary relationships between species. Occasionally, mutations provide benefits to an organism. These beneficial mutations can lead to new traits that improve survival or reproduction. For example, a mutation that confers resistance to a disease can spread through a population over time. Beneficial mutations are a driving force behind adaptation and evolution, shaping the diversity of life. Mutations are harmful, disrupting normal gene function and leading to diseases or developmental problems. These mutations can be inherited or acquired and may result in conditions like cystic fibrosis, muscular dystrophy, or certain cancers. Understanding harmful mutations is essential for developing treatments and preventive strategies. Genetic drift is a process that can change the frequency of polymorphisms in a population over time, especially in small populations. Unlike natural selection, genetic drift is random and can lead to the loss or fixation of certain genetic variants. This process contributes to the genetic uniqueness of isolated populations and can influence the prevalence of certain traits. Polymorphisms often serve as genetic markers, helping scientists track inheritance patterns and identify genes associated with diseases. These markers are invaluable in genetic mapping, forensic analysis, and ancestry research. By analyzing genetic markers, researchers can trace lineage, study population history, and improve disease diagnosis. Population genetics studies the distribution and changes of genetic variations, including mutations and polymorphisms within populations. This field helps explain how evolutionary forces like selection, drift, and migration shape genetic diversity. By analyzing genetic data, scientists can understand disease prevalence, adaptation, and the evolutionary history of populations. Genetic testing uses information about mutations and polymorphisms to assess disease risk, diagnose conditions, and guide treatment. Tests can screen for specific mutations linked to inherited diseases or analyze polymorphisms that affect drug response. As technology advances, genetic testing is becoming more accessible and informative, empowering individuals to make informed health decisions. Gene therapy aims to treat or prevent diseases by correcting or replacing faulty genes. This innovative approach often targets specific mutations responsible for genetic disorders. By delivering healthy copies of genes or editing the genome directly, gene therapy holds promise for curing previously untreatable conditions. Ongoing research continues to expand its potential applications. Study and application of mutations and polymorphisms raise important ethical questions. Issues include privacy, consent, and the potential for genetic discrimination. As genetic information becomes more integrated into healthcare, it's crucial to establish guidelines that protect individuals' rights and ensure responsible use of genetic data. Personalized medicine uses genetic information, including mutations and polymorphisms, to tailor healthcare to each individual. By understanding a person's unique genetic makeup, doctors can select the most effective treatments and avoid those likely to cause side effects. This approach is revolutionizing healthcare, making it more precise and effective. Mutations and polymorphisms are also important in agriculture. Plant breeders use natural and induced mutations to develop crops with desirable traits, such as disease resistance or improved yield. Understanding genetic variation helps create more resilient and productive plants, supporting food security and sustainability. Animal breeding also benefits from knowledge of mutations and polymorphisms. By selecting for beneficial genetic variations, breeders can enhance traits like growth rate, disease resistance, or milk production. Genetic testing helps identify animals with desirable genes, improving breeding programs and animal health. Environmental factors can influence the rate and type of mutations in living organisms. Pollution, radiation, and chemicals can increase mutation rates, sometimes leading to negative health effects or loss of biodiversity. Monitoring and managing environmental risks is essential for protecting both human health and ecosystems. Mutations and polymorphisms are the raw material for evolution. They introduce genetic diversity, allowing populations to adapt to changing environments. Over time, beneficial variations become more common while harmful ones may disappear. This ongoing process shapes the evolution of species and the diversity of life on Earth. Polymorphisms are a major source of human diversity. They explain differences in physical traits, susceptibility to diseases, and even how we perceive tastes or smells. By studying these variations, scientists gain a deeper understanding of what makes each person unique and how populations have adapted to different environments. Genetic counseling helps individuals and families understand the implications of mutations and polymorphisms. Counselors provide information about inherited conditions, disease risks, and testing options. This support is invaluable for making informed decisions about health, family planning, and managing genetic disorders. Genetic engineering allows scientists to introduce, remove, or modify genes in organisms. By harnessing knowledge of mutations and polymorphisms, researchers can create genetically modified organisms, GMOs, with specific traits. This technology has applications in medicine, agriculture, and industry, offering solutions to global challenges. CRISPR is a revolutionary genome editing tool that allows precise changes to DNA. Scientists use CRISPR to correct mutations, study gene function, and develop new therapies. This technology has transformed genetics, making it possible to edit genes with unprecedented accuracy and efficiency. Epigenetics refers to changes in gene expression that do not involve alterations in the DNA sequence itself. However, mutations can influence epigenetic mechanisms and vice versa. Understanding the interplay between mutations and epigenetics is crucial for unraveling complex traits and diseases. Large genetic databases store information about mutations and polymorphisms from diverse populations. These resources help researchers identify disease-associated variants, study population genetics, and develop new treatments. The growth of genetic databases is accelerating discoveries in medicine and biology. Bioinformatics combines biology, computer science, and statistics to analyze genetic data. By processing vast amounts of information about mutations and polymorphisms, bioinformatics enables scientists to identify patterns, predict disease risk, and develop new therapies. This field is essential for modern genetic research. The future of genetic research is bright, with new technologies and discoveries on the horizon. As we learn more about mutations and polymorphisms, we'll unlock new ways to prevent, diagnose, and treat diseases. Continued research promises to improve health, enhance agriculture, and deepen our understanding of life itself. Despite remarkable progress, challenges remain in genetics. Interpreting the effects of mutations and polymorphisms is complex, and not all genetic changes are well understood. Ethical, legal, and social issues must also be addressed as genetic information becomes more widely used. Ongoing collaboration and dialogue are essential for responsible advancement. Educating the public about mutations and polymorphisms is vital for informed decision-making. Clear communication helps dispel myths, reduce stigma, and promote acceptance of genetic diversity. As genetics becomes more integrated into daily life, public understanding will support responsible use and innovation. Genetic literacy means having the knowledge and skills to understand genetic information. As genetic testing and personalized medicine become more common, improving genetic literacy is essential for individuals to make informed health choices and participate in discussions about genetics in society. Let's recap what we've learned. Mutations and polymorphisms are changes in DNA that drive genetic diversity. Mutations can be rare and impactful, while polymorphisms are common and usually harmless. Both play essential roles in evolution, health, and disease. Advances in genetics are transforming medicine, agriculture, and our understanding of life. By staying informed and considering ethical implications, we can harness the power of genetics for the benefit of all. As we look to the future, the study of mutations and polymorphisms will continue to shape science, medicine, and society. With ongoing research and technological advances, we'll gain deeper insights into our genetic makeup and how it influences every aspect of life. Thank you for joining us on this journey through the fascinating world of genetic variation. Stay curious and keep exploring.